happens in a bear to a whale. It occurs in all life and is responsible for all of us. It is mitosis and meiosis.
Oh, hello! I didn't see you there. Reading my favorite novel, 1001 Cool Schoolyard Jokes. It's my favorite excerpt. Why did the lion feel sick after he'd eaten the priest? Because it's hard to keep a good man down. Yes it is. But now, let's talk about a different topic, not of the humorous content. Mitosis. Mitosis happens in every cell excluding sex cells. It is the process of splitting the nucleus, and the cell's ability to split is due to the copying and separation of the nucleus. And before we get into mitosis, a crucial point is interphase. Interphase is the period of cell growth, and this is when the cell makes new molecules and organelles and carries out normal functions such as cellular respiration. The centrosomes are duplicated but cannot be distinguished individually because they are in the form of chromatin fibers and not have yet coiled around histones. The chromatin duplicates itself in the nuclear membrane, and so does the centrosomes, preparing for the start of mitosis called prophase. In prophase, the nuclear envelope is destroyed and the chromatin fibers coil around histones. And now they are called chromosomes and can be seen by a microscope. Two identical sister chromatids are joined by a centromere, kind of at the waist. Microtubules grow from the centrosomes that are moving towards opposite sides of the cell and attach to the centromeres, which push the chromosomes toward the center of the cell. Metaphase. The mitotic spindle has reached the maximum growth, and the microtubules on the centromere of each sister chromatid each pull them towards the centrosomes. This makes a tug-of-war effect of each side trying to get the chromosomes. The motor proteins attached to the microtubules spool it and pull the chromosomes toward the center of the cell. Anaphase. This is when the sister chromatids split, and now two daughter chromosomes that are pulled to opposite sides of the cell as the microtubules shorten. The microtubules that were not attached to the centrosome lengthen, which makes the cell longer. Dynein, a protein, plays tug-of-war with itself to make the two sister chromatids split. Once the chromosomes have reached opposite sides of the cell, the cell nuclear envelops to form around them, and they uncoil into chromatid once again, and the mitotic spindle disappears. And there is a cleavage furrow in the middle as the cell is pinched into two identical daughter cells, and that's mitosis. Stick around, maybe you'll see meiosis.
Sorry. Thank you, Bartholomew. I appreciate it. Meiosis. Meiosis is when one diploid cell splits into 4 unique haploid cells. And a diploid cell is 46 chromosomes, and a haploid cell is 23 chromosomes. And this happens twice. Before they can go through meiosis, first the cell must go through interphase. Interphase is the period of cell growth, and this is when the cell makes new organelles. The centrosomes are duplicated, which are proteins which basically tell everything where to go in the cell. And the DNA cannot be distinguished individually because they are in the form of chromatin fibers, which are also duplicated inside the nuclear membrane. Because we are splitting twice, there's going to be prophase. Of every stage, there's going to be a number 1 and a number 2. And prophase 1 is the same as mitosis except for crossover and homologous recombination. There are two X's, or a double chromosome. There's one X that is entirely of your mother's next to one that is entirely your father's. And they're each the same homolog, which means there's the same trait. The four chromatids cross over and they get tangled up in trade sections of DNA. That's homologous recombination. The actual part of getting tangled up is called crossover. This happens entirely in all chromosomes except for male sex chromosomes, in which the X of female will refuse to bond with the Y chromosome. The parts of the thing are trading from the same location, and it creates unique gene combinations. This is the main component for natural selection, which is variation, and one of the reasons we're able to adapt. One of each unique chromosome will end up in 4 of the different cells. Metaphase 1. Each homologous pair migrates to either end of the cell, and all the chromosomes line up with their homologous pairs. Anaphase 1 is the exact same as mitosis, with the microtubules causing them to split, the dynein protein, and the motor proteins. Telophase 1. Now there are 2
unique haploid cells, which each will be split again in all of the stages coming up. Prophase 2
is the exact same as prophase 1, except for one big thing. There is no crossover. There is no DNA replication involved, so only half of the normal 46 chromosomes will go into each cell. Metaphase 2, all the chromosomes are moved into alignment in the center of the cell. Anaphase 2, all the chromosomes are pulled apart into separate and single chromosomes. Keep in mind that for everything that has 2 in it, there are 2 cells doing this, not 1. Telophase 2, the 2 cells split, and now there are 4 unique cells, each with 23 chromosomes. So there are 4 haploid cells.
Process is different. First, the end result of mitosis are 2 identical daughter cells, which are diploid, and in meiosis, the end result are 4 unique haploid cells. Also, meiosis is only used to create gametes, which are eggs and sperm, while mitosis is used in almost all the cells in the body. Why is it important? Meiosis allows sexual reproduction, which creates more variation among organisms. It also creates 4 unique haploid cells, which creates even more variation. And variation is important because it allows organisms to change over time and become more fit in an environment due to natural selection. Mitosis is important because it allows organs to replenish and heal themselves. It also allows organisms to decrease in size without the cells dying from becoming too large and not being able to get enough nutrients and oxygen.